My name is Yamilka Nunez, and I will be showing the melanocortin 1 receptor, also known as MC1R, and the mutation shown is arginine number 67 being replaced by cysteine number 67. The normal function of MC1R is to regulate skin and hair pigmentation by controlling the production of melanin in melanocytes. I would categorize MC1R as a receptor because once it binds, the protein stimulates loss of your melanin in order to have a high production of pigmentation. The receptor's subcellular location is the plasma membrane, but is specifically found on melanocytes. MC1R is found in vertebrate organisms such as birds, humans, mice, and horses. The protein becomes less active after the mutation is added onto the protein because when you compare the light-colored mice with the dark-colored mice, the location of arginine in dark-colored mice being replaced by cysteine is shown through the light-colored mice, which resulted in less melanin being produced, showing that there was less stimulation of the MC1R protein. The main functional groups present in the side chain is 2 amino groups and CH2. It is hydrophilic and positively charged, along with it being large compared to other standard amino groups. The amino groups in the original side chain can accept hydrogen ions and eventually form NH2, allowing NH2 to interact with water and produce more NH2 molecules. The main functional groups present in the mutated side chain is sulfonyl hydro groups and a CH2 molecule. It is also hydrophilic, but the charge is neutral, and it is small compared to the other 19 amino side chains. The mutated amino side chain produces interactions like disulfide bonds formation due to cysteine now being placed next to another cysteine. The mutation also leads to altered redox sensitivity, which can lead to a change in how the receptor interacts with ligands or how it responds to redox signals. In other words, the receptor is not activated as much as how it's activated with the insert of arginine instead of cysteine. The assigned mutation affects the protein function because it interferes with the stimulation of the protein, causing less eumelanin to be produced, meaning less melanin will be expressed in the organism. The assigned mutation can impact the function of the cell because it will destabilize the structure of the receptor, not allowing it to bind, meaning the receptor won't be activated. The assigned mutation does not necessarily impact the function of the organism, but the only impact is that the lack of melanin produced can impact the organism's survival. For example, light-colored mice in dark-colored environments are more visible to predators, so they will have to find more strategic ways to survive compared to dark-colored mice. My model is in the shape of a chain, which I showed by bending the purple stems. In the mutated protein, I show how cysteine replaces arginine by using a different colored bead than the bead I used in the original protein. The 2 yellow lines represent the cell membrane, which is where the protein is located, and the white beads represent the amino acids. One limitation I had was that I wasn't able to show how there was a reduced amount of melanin produced in the mutated protein because of lack of materials. Another limitation I found was that I wasn't able to label all the functional groups in the proteins and how it shifted in the mutated protein. Another limitation I had was that I wasn't able to show how MC1R binds to the melanocyte, stimulating the protein and producing more pigmentation.